A pleasant day to everyone, most especially to our panelists, Mr. Lester Torres, Mrs. Rosana Arelia, and last but not the least, Mrs. Leonila Santos. Today we are here to present you our experimental research study guided by Mr. Kaisit and Mrs. Santos entitled The Lantern Leaves and Peelings, its efficiency as natural hand sanitizer against Staphylococcus aureus and Escherichia coli. We have here the people behind the paper who were able to accomplish this study. I'm Shin Galvez,
Bernardino Nicole, Althea Santos, Mingi Chamuel Rain, T.C. Luis Jansen, Peñalosa Carl Timothy.
and that concludes the members of Group 4. Here are the overview of our presentation. This study aimed to create a hand sanitizer out of Dalandan leaves and peelings. Alcohol-based hand sanitizer are more effective at killing microorganisms than soaps and do not dry out hands as much. Citrus has high levels of acidity where it is used as a cleaning material in indoor areas because it contains limonene. It also gives off a pleasant odor. That is why citrus is one of the most efficient and effective that has the potential to be sanitizer due to its ability to remove dirt, including killing various types of bacteria. The acidity contained in the citrus is called citric acid. Based on the study, it is a weak acid found naturally in all citrus fruits.
Hi, I am Nicole Bertandino and I'm going to discuss the theoretical framework. Two flavonoids, naringenin and hesperidin, have been proven to have anti-inflammatory and anti-heterogenic characteristics according to Hervanix 2018. Dalandan has antiseptic, antifungal, antiviral, muscle pain reliever, and edema, rheumatism, anti-aging, and antibacterial activities. And there is no scientific evidence that bitter orange improved wound healing according to Hello Doctor 2021. Because Dalandan is high in vitamin C, it is safe to assume it will help with wound healing. Vitamin C, on the other hand, is essential for the formation of collagen, which is required for the healing of skin damage.
Figure 1.0, the conceptual framework of the study. The conceptual framework of the study as shown above represents its essential aspects, including the dependent and independent variables, as well as the processes involved. The researchers produced an organic hand sanitizer made from Dalandan from its remains such as leaves, peels, and seeds. Dalandan reviewed both commercially directed and experimentally produced goods, and the results were subjected to laboratory testing and feedback from the respondents. Antimicrobial activity against S. aureus and E. coli and portable pH level meter test were among the lab testing procedures. The respondents' judgment examined nutritious effects, relaxing or soothing results, smell, fragrance, and consistency. Finally, the information acquired from these tests and evaluations was analyzed and compared.
The statement of the problem determines the efficacy of Citrus X aurantium L Dalandan leaves and peelings as an alternative hand sanitizer. Listed here are the problems as follows. Number 1. What were the formulations used in making the product DLPHS or Dalandan leaves peelings extra hand sanitizer and SCHS or commercial hand sanitizer? Number 2. What are the pH levels of DLPHS and CHS? Number 3, what were the results of the antimicrobial activity test for the DLPHS and CHS against the bacteria Escherichia coli and Staphylococcus aureus? Number 4, what were the results of the evaluation of the respondents regarding the designated qualities of DLPHS and CHS in terms of fragrance, color and appearance, texture and viscosity, and soothing effects?
Null hypothesis. Number 1. There is no significant change in the fragrance of citrus, ex aurantium, and dalandan leaves and peelings on the alcohol. Number 2. It is not effective in eradicating a wide range of bacteria, germs, and pathogens. Alternative hypothesis. Number 1. There is a significant change in the fragrance of citrus, ex aurantium, and dalandan leaves and peelings on the alcohol. Number 2. It is effective in eradicating a wide range of bacteria, germs, and pathogens.
Now let's move on over to the significance of the study. Significance of the study. The research conducted is essential because it will benefit the following people. Number 1, to the students. The study is significant for students because it enables them to determine what alternatives they can use if they run out of hand sanitizers. It is also less expensive than the commercial hand sanitizers sold in the market. This research may also help students in implementing environmental wellness. Second, to the teachers. The findings can serve as basic knowledge related to antiseptics. It may help them identify and assess the capability of dalandan, which may guide them to become more effective educators. The result of the study can give an idea and environmental awareness. Next up, to the manufacturers. The study benefits manufacturers by promoting organic disinfectants and sanitizers that are made healthily and naturally. It is chemical free and non-toxic to humans, making it suitable for use in food preparation areas. To the residents, the study will benefit the residents in diminishing waste production and utilizing it as a hand sanitizer to decrease the amount of waste for disposal, save space in landfills, and conserve natural resources. And finally, to the future researchers, the study will eventually benefit future researchers regarding the ability of a dalandan fruit to be used for a homemade hand sanitizer. It will undoubtedly help future researchers analyze and expand the other types of content about making hand sanitizers with different classifications of fruits.
The next category that I will be presenting is the scope and delineation. This study aims to make an antibacterial hand sanitizer out of dalandan peels and leaves, as well as to compare the aroma, consistency, affordability, and efficiency of commercial and dalandan hand sanitizers. The research was conducted at the researchers' homes and the other laboratory testing facilities. The researchers obtained the mixture using extraction and macellation methods to gather observations and feedbacks. The researchers tested it on 30 respondents in their population. This study concluded and conducted at Rodriguez Rizal in the Philippines. Due to their numerous antimicrobial testing, experimenting, creating product, and writing the research paper, this study took 9 months.
Definition of terms. The following terms were defined operationally as they are used in the process of this research. Citrus ex aurantium L. dalandan refers to the scientific name of the fruit bitter orange, which was used by the researchers to come up with an alternative hand sanitizer. Coronavirus COVID-19 refers to a group of RNA viruses that cause a variety of respiratory, gastrointestinal, and neurological disease in humans and other animals, which could possibly be avoided with the use of the alternative hand sanitizer. D-limonene refers to the chemical found in the ribs of citrus fruits such as lemon, limes, and oranges. The diffusion method, DTM, is classified as an agar diffusion method, ADM, because the plant extract to be tested diffuses from its reservoir through the agar medium seeded with the test microorganism. Glycerin, it is a thick, sweet, colorless liquid that is used especially in making medicines, explosives, and antifreeze for cars. Limonene, it is the major component in the oil of oranges, which has many uses including as flavor and fragrance. Inhibitory activity, it is the phenomenon in which is compound called an inhibitor, in most cases similar in structure to the substrate upon which an enzyme acts to form a product and react with the enzyme so that the resulting complex either cannot undergo the usual reaction or cannot form the usual product. pH level is a measure of how acidic base water is. Ocacillin is a semisynthetic penicillin administered in the form of its hydrated sodium salt to treat infection caused by penicillin-resistant Staphylococcus. Amikacin is an aminoglycoside used to treat infections caused by more resistant strain of gram-negative bacteria and some gram-positive bacteria. Soothing effect, the effect of which that makes someone feel calm and relaxed. Texture, the feel, appearance, or consistency of a surface or substance. Fragrance, a pleasant or sweet smell usually correlated with the smell of flowers or flowery scent. Color, the property possessed by an object or producing different sensation on the eye because of the way the object reflects or emits light. Staphylococcus aureus, recognized as one of the most important bacteria that cause disease in humans. It is the leading cause of skin and soft tissue infections such as ovositis, boils, furuncles, and cellulitis. Escherichia coli are bacteria found in the environment, food, and intestine of people and animals. E. coli are a large and diverse group of bacteria. DMPHS, the London Leaves and Peelings Extract, hand sanitizer. This refers to the product made by extracting the dalandan leaves and peel extract by the researchers. CHS, commercial hand sanitizer. This refers to the product that is available to the market and is mass produced.
Now, let's move on over to the research design and methodologies. Methods of research. Experimental research is a scientific method of doing research in which independent factors are altered and applied to one or more dependent variables in order to determine their influence on the latter. The influence of the independent variables on dependent variables is often observed and recorded over time to assist researchers in reaching a reasonable conclusion about the link between these two variable types. Because of our research on the extraction of dalandan peels, the experimental research was best matched. The effect of the independent variables on the dependent variables is usually observed and recorded over some time to aid researchers in drawing a reasonable conclusion and regarding the relationship between these two variable types. Moreover, this is partly due to the common misconception that the research is equivalent to scientific experiments, a component of experimental research design.
Respondents of the study. Due to the presence of pandemic COVID-19, the respondents of the study contained only 30 people. These are the individual people who came from the neighborhood of the researchers in the various barangays of Rodriguez Rizal. Every researcher will take hold of 5 respondents with student, teacher, resident, manufacturer, and lastly, future researcher. The 6 researchers selected family from their community. The total is 30 individual people. The researchers used purposive sampling techniques in selecting the respondents, indicating that these individuals were selected for research because they met a certain criterion.
For the research instrument, the researchers gathered the necessary data and used survey questionnaires. In the preparation of the instrument, the requirements in the designing of good data collection instruments were considered. The researchers have few questions for the respondents through the use of survey questionnaires. These questionnaires were provided to the respondents to make an evaluation or feedback on the products. The question or surveys provided were validated before being administered to the respondents to ensure an effective data gathering procedure.
Data collection procedure. The researchers began their investigation with the goal of discovering an interesting and relevant research study topic. The researchers initially sought approval from the research advisor, Mr. Rafael Kaisip, after deciding on the research title. The researcher also obtained permission from FE Roosevelt Campus President, Mrs. Maria Teresa S. Angeles, for granting permission to perform the study. The researchers began developing the goods that they would utilize in the trial after the permit was approved. The researcher would begin by obtaining the necessary supplies and ingredients for their creations, and the majority of which were ordered online because to the ongoing COVID-19 pandemic constraints. Following the creation of product samples, the researcher assessed the samples and selected a sample from which the product would be sent to a laboratory for further testing. The researcher would then choose which test would they perform on their product, eventually settling on antibiotic activity test against Staphylococcus aureus and Escherichia coli. The researchers awaited the outcomes.
Moving on, let us now discuss the statistical tools and treatment of data. Number 1, the efficiency formula. This formula was used to determine the pH value rates of the DLPHS and CHS. Number 2, the optimal pH rate range formula. This was used to determine whether the DLPHS and CHS match the optimal values of disinfectant sprays. Number 3, the weighted mean. The formula was used to calculate the average evaluation of the 30 respondents regarding the responses to the products DLPHS and CHS that they were instructed to use. Number 4, the t-test. The formula was intended to give evidence that would either accept or reject the null hypothesis of the study. Formulas required for the one-tailed paired samples t-test. Number 1, which is the computed t-value. Number 2 would be the degree of freedom, and number 3 would be the confidence interval of difference.
Now, moving on to Chapter 4 presents, analyzes, and interprets the data obtained by the researchers.
Table 1.1 presents the features of the London Leaves peelings. Extract hand sanitizer and commercial hand sanitizer consist of color, fragrance, texture, viscosity. On the London Leaves peelings extract hand sanitizer, the color appears to be lime green, whereas on the commercial hand sanitizer, it appears to be clear and translucent. The London Leaves peelings extract hand sanitizer has an enticing citrus aroma, whilst the commercial hand sanitizer has a strong alcohol scent. The texture and viscosity of the London Leaves peeling extract hand sanitizer appear to be much more watery and runny, whereas commercial hand sanitizer looks to be thick and viscous to touch with. Table introducing the London Leaves Peelings Extract Hand Sanitizer and Commercial Hand Sanitizer. It was illustrated above that quantities each component had its percentage to create an 885 ml the London Peel and the London Leaves Extract Hand Sanitizer. The procedures product formulation were derived from the product percentage composition as recommended in various related literature and studies. In the product formulation, the concentration of ethanol shown in the various studies was followed. The researchers used the London extract from the London peels and the London leaves as their main ingredients, including ethanol, which has the ability to kill germs and bacteria. The function of the thickening agent is distinctly based on the name itself. It was used for the thickeners of the sample. And lastly, distilled water was used for the extraction of the citrus.
Table 2.1. Disk diffusion test results of Escherichia coli with DLPHS and CHS. In an antimicrobial activity test, positive and negative controls are always present. Positive controls are tools that usually serve as a comparison. Meanwhile, negative controls serve as a tool that does not bring any changes whatsoever to the experiment. Furthermore, it is usually used to control unknown variables during experimentation and to check if there were no contaminations present in the experimental setup. The antimicrobial activity test results showed that both sample products produced complete inhibitory activity against the bacteria Escherichia coli with mild reactivity against the previously mentioned test organism. Table 2.2. Disk diffusion test results of Staphylococcus aureus with DLPHS and CHS. Table 2.2 presents the antimicrobial activity of DLPHS and CHS against Staphylococcus aureus. The antimicrobial activity test results showed that both samples produced partial inhibitory activity with mild reactivity against the test organism, Staphylococcus aureus. Additionally, the oxacillin served as the positive control, producing complete inhibitory activity with severe reactivity against the test organism. In conclusion, the antimicrobial activity test results show that the DLPHS is just as effective as the CHS in terms of preventing bacterial growth and propagation.
Table 3.1 illustrates the measured pH value of the London Leaves Peeling Extract Hand Sanitizer and Commercial Hand Sanitizer. These measurements were obtained by using a pH meter as shown above. In the figure, the London Leaves Peeling Extract Hand Sanitizer has a pH value of 4.00 and Commercial Hand Sanitizer had a pH value of 9.80. The acidic nature of the London extract may be accounted for the decrease in pH level since the pH level of most fruits containing citric acid usually range between 3 to 6. An acidic hand sanitizer could help in removing more stains, according to Kogel 2012 acids. Acids, more specifically citric acid, have the compounded property to efficiently remove stains from clothes. It also has the capacity to kill germs. Normal alcohol-based hand sanitizer usually can't kill, meaning the nature of the London Leaves Peeling Extract Hand Sanitizer being acidic makes it very good and efficient. Table 3.2 compares the measured pH value of the London Leaves Peeling Extract Hand Sanitizer and Commercial Hand Sanitizer to the optimal pH range of a standard hand sanitizer. The shown pH level rates of the values were obtained from the ratio of this value to the maximum optimum pH value 10.5 multiplied by 100%. As indicated above, the pH level rate of the London Leaves Peeling Extract Hand Sanitizer is 38.09% optimal rate range, while the pH level rate of commercial hand sanitizer is 87.42%, is beyond the optimal rate range. This excess rate has negligible effects for most skin types but may be found considerable for those with sensitive skin conditions.
Table 4.1 shows the gender and age distribution of students who responded. In terms of gender, there are 17 females who participated, representing 56.67% of the total respondents, while there are 13 males, which equates to 43.33%, which result in a total of 30 respondents. Furthermore, the table depicts the respondents' ages. There are 13 respondents who are 13 to 19 years old, representing 43.33% of the total respondents. 7 respondents who are 20 to 29 years old, which result in 23.33% of the respondents. 2 respondents who are 30 to 39 years old, representing 6.67% of the respondents. 6 respondents who are 40 to 49 years old, representing 20% of the respondents. And 2 respondents who are 70 to 79 years old, representing 6.67% with a total of 30 respondents. Table 4.2 presents the professional status of respondents who responded. The table shows the student, teachers, residents, future researchers, and manufacturers. In total, there are 11 students with percentage of 36.67%, 5 teachers with a percentage of 16.67%, 9 residents with percentage of 30%, 1 manufacturer with a percentage of 30.33%, and 4 future researchers with a percentage of 13.33%, totaling 30 respondents.
Table 5.1. Evaluation of the respondents regarding the indicated qualities of DLPHS and CHS in terms of fragrance. Table 5.1 shows the preference of the respondents on the indicated quality of DLPHS in terms of fragrance. With a weighted mean of 3.9, 3.47, 3.83, respectively, respondents agreed that DLPHS as an appealing scent lasted a long time and its actual scent originated from its ingredients. On the other hand, their scores are neutral with a weighted mean of 3.23 in terms of DLPHS having a unique smell. Meanwhile, respondents agreed that the CHS has an appealing scent, that it has a distinct scent, that it has a long-lasting scent, and that its actual scent is derived from its ingredients, with a weighted mean of 3.79, 3.52, 3.83, and 3.62, respectively, based on the indicated average means of DLPHS and CHS. The respondents agreed that both products contain the previously mentioned qualities. Table 5.2. Evaluation of the respondents regarding the indicated qualities of DLPHS and CHS in terms of color and appearance. Table 5.2 shows the respondents' evaluation on the color and appearance qualities of DLPHS and CHS. The respondents were neutral about the DLPHS having an appealing color with a weighted mean of 3.23. However, the respondents strongly agreed that the DLPHS did not leave any color stains with a weighted mean of 4.2. Meanwhile, respondents also felt that the CHS had a pleasing color and did not leave any color stains, with a weighted mean of 3.83 and 4.17 respectively. Based on the indicated average means of DLPHS and CHS, the respondents agreed that the both products contains the previously mentioned qualities. Table 5.3. Evaluation of the respondents regarding the designated qualities of DLPHS and CHS in terms of texture and viscosity. Table 5.3 illustrates how the respondents rated the texture and viscosity of DLPHS and CHS. Meanwhile, with a weighted mean of 3.97, the respondents agreed that the CHS had a non-sticky consistency. Furthermore, the respondents strongly agreed when it came to the CHS not having a rough or lumpy texture, with a weighted mean of 4.27. Based on the stated average means, the respondents agreed that DLPHS, CHS contained the previously mentioned qualities. Table 5.4 Evaluation of the respondents regarding the designated qualities of DLPHS and CHS in terms of soothing effects. The respondents' ratings, the defined qualities of DLPHS and CHS in terms of soothing effects, are shown in Table 5.4. The respondents felt that the DLPHS having a relaxing and comforting experience, as well as hydrating and moisturizing effect, with a weighted mean of 3.83 and 3.97. Meanwhile, with a weighted mean of 4.03 and 4.13, respondents agreed that the CHS produced a relaxing and comforting experience as well as a hydrating moisturizing effect. Based on the indicated average means of the DLPHS and CHS, the respondents agreed that both products contain previously mentioned qualities.
Table 6 shows the respondents' preferred hand sanitizer and the percentage of respondents who chose it based on color, aroma, texture, and relaxing effects. In terms of color, as indicated above, 20 respondents chose the London Leaves Extracts hand sanitizer with 67%, while 9 respondents chose commercial hand sanitizer with 30%. As a result, respondents favored the London Leaves Extract hand sanitizer over commercial hand sanitizer in terms of color since it was more appealing and it did not leave any stains after use. Furthermore, in terms of aroma, 25 respondents of 83% preferred commercial hand sanitizer, whereas 4 respondents of 13% chose the London Leaves Extracts hand sanitizer. As a result, respondents favored the London Leaves Extracts hand sanitizer over commercial hand sanitizer in terms of scent, since it had a pleasant smell that lingered after it was used. The components were identified by their scent.
Table 8.1 indicates the significant difference in the evaluation of the respondents to the indicated qualities of the London Leaves Peeling Extract hand sanitizer and commercial hand sanitizer in terms of color and appearance. Since the computed t-value of negative 0.93 was less than the critical t-value of 1.67 and did not fail into the rejection region, the null hypothesis was accepted. This implied that there was no significant difference in the evaluation of the respondent to the indicated qualities of the London Leaves Peeling Extract hand sanitizer and commercial hand sanitizer in terms of color and appearance. Table 8.2 indicates the significant difference in the evaluation of the respondents to the indicated qualities of the London Leaves Peeling Extract hand sanitizer and commercial hand sanitizer in terms of fragrance, since the computed t-value of negative 0.43 was less than the critical t-value of 1.67 and did not fail in the rejection region, so the null hypothesis was accepted. This implied that there was no significant difference in the evaluation of the respondents to the indicated qualities of the London Leaves Peeling Extract hand sanitizer and commercial hand sanitizer in terms of fragrance.
Table 8.3 demonstrates the significant difference between the outcomes of the respondents' evaluation of DLPHS and CHS in terms of texture, since the computed t-value of 0.80 was less than the critical t-value of 1.67 and did not fall in the rejection region. Thus, the null hypothesis was accepted.
Table 8.4. Significant difference between the result of evaluation of the respondents on the indicated qualities of DLPHS and CHS in terms of its suiting effects. Table 8.4 displays the significant difference between the outcomes of the respondents' evaluation in the indicated qualities of DLPHS and CHS in terms of suiting effects. The decision was to accept the null hypothesis because the computed t-value of negative 1.26 was less than the critical t-value of 1.67, and it did not fall in the rejection region. Therefore, there was no significant difference in the evaluation of the respondents to the indicated qualities of DLPHS and CHS in terms of its suiting effects.
Moving on to the fifth chapter, conclusion and recommendation from the researchers.
Based on the findings of the study, the following conclusions were drawn, such as the number 1, the formulation used for the product DLPHS were concluded to have a natural and appealing scent compared to CHS. Number 2, the pH level measurements of the two products, such as the DLPHS with a pH value of 4% and the CHS with a pH value of 9.18%. Number 3, based on the antimicrobial activity test results, it is inferred that the DLPHS is just as efficient as the CHS. Number 4, based on the results of the evaluation regarding the indicated qualities, the respondents agreed on DLPHS and CHS in terms of fragrance, color and appearance, and suiting effects. One thing to note is the respondents strongly agreed with the CHS texture and viscosity. Lastly, number 5, based on the results of the hypothesis test, it was observed and concluded that there was no significant difference in the indicated qualities of DLPHS and CHS in terms of fragrance, color and appearance, texture and viscosity, and suiting effects.
Recommendations. Based on the conclusions drawn, the following recommendations were given by the researchers. To the general public, they are recommended to use this product as an alternative in the case that commercial hand sanitizers become unavailable or unobtainable. Furthermore, the findings of this study help back up its viability to serve its purpose as an alternative hand sanitizer that could save the money and keep them safe during the pandemic. To product manufacturers, they are recommended to use this product and findings of this study to furthermore investigate alternative hand sanitizer solutions that can possibly be made even cheaper and more accessible to the public. Moreover, it can be promoted as a sort of organic and more natural sanitizing product. To future researchers, they are recommended to use the product and the findings of this study to focus more on the efficiency of which alternative hand sanitizers kill bacteria. They are recommended to do more tests like the aerobic plate, count test, etc. to have a better understanding on how to make an alternative hand sanitizer solutions and to provide more insight regarding the viability of alternative hand sanitizers.
Thank you for listening to our presentation and paying attention to our topic.